Assalamu alaikum wa rahmatullahi wa barakatuh. This playlist is going to be in English. These are revision videos on embryological development or EMD. It's been a long time that I've last made an English video, and since it's not my first language, you may witness some mistakes in terms of language, but it's not an obstacle. The communication will be improved throughout the videos, inshallah. Anyway, we are revising embryology, and embryology is the study of the fetus. It's divided into two main sections. The first section is the general embryology, and the second one is the systemic, also called the special embryology. And this is to be taught later, inshallah. But now our concentration on the general embryology. And I emphasize that it will be just revising videos. I'll give you the summary, and then if I can, I'll be making questions videos as well. I prefer to explain directly from the book or making summaries on white paper. So you just need to follow up with me. The first chapter or the first topic we are going to start in the general embryology is gametogenesis. The word genesis means production, and the word gamete refers to either sperms or ova. Sperms from the male and ova from the female. So this first chapter of the general embryology is going to talk about spermatogenesis first, sperms production, followed by oogenesis, and that's in the next video, inshallah. So be ready with our first video on the spermatogenesis, بإذن الله.
يخلقكم في بطون أمهاتكم خلقا من بعد خلق في ظلمات ثلاث.
Bismillah ar-Rahman ar-Rahim. So what does the word spermatogenesis means? And what are we going to study about it? Well, first of all, we already mentioned that genesis means production. So here we are producing the sperms. There are steps that you have to be having in mind first before we start reading the book. For the sperms to be produced, or their other name, spermatozoa, steps has to occur and transformation. We first start by a cell called spermatogonia. Then it's transformed into spermatocyte, and then transformed into spermatid, and lastly into spermatozoa. You should be noticing that the first part of these four words starts with the prefix spermato, referring to the sperm's production process. And then the second part, each one has its own name. Gonia here refers to a germ cell, and cyte means a cell, and here zoa means a living substance. I recommend you stop the video at this moment and write these four in a column like this. You stop the video and written them. Okay, let's continue. Here is the PDF. I'll be leaving in the comments below of the first chapter of the embryology. We are starting with the spermatogenesis. We will study with each other or revise the definition, then the site and the onset, followed by the stages of the spermatogenesis, and then some data or statistics. Only these 5 titles. Starting with the definition. We already said that spermatogenesis simply means the production of the sperms, or their other name spermatozoa. From where? From our first cell, the spermatogonia. If we returned here to the summary, this is the summary whiteboard. Our first name that we did memorize is the spermatogonia. During its production process, the last product is the spermatozoa or the sperms. So the definition is now clear. It is the process by which spermatogonia transferred into spermatozoa. Where it occurs? Well, we are talking about sperms, so it has to be in the testis of the male, and that's its site. Where exactly? In tubules, we are going to study later, called seminiferous tubules. Tubules means small tubes, and seminiferous also divided into 2 parts. The word ferrous in Greek or Latin means producing. So these are tubules that produce the semen. They are the functional units of the testis. And the onset simply starts at the puberty until that male dies. Unlike with the females, until menopause. Okay, definition, site, and onset we read about. The stages are 4. Reminding ourselves that we are revising, we can remember the stages in a mnemonic, which is the word pigment. Proliferation, then growth, then maturation, then transformation. Proliferation simply means dividing. The spermatogonia will divide to increase in the number, and then grow to increase in the size, forming the spermatocytes. Just as we said, the spermatogonia proliferate and grow to form the spermatocyte. For more details, let us read. Spermatogonia, just like any cell in our body, has diploid number of chromosomes, meaning it has diplo in Greek means double. It has double the sets of chromosomes. N here means one set of chromosomes, and N equals 23 in humans. So 2 multiplied by 23, since we're talking about double here, means 46. These are 46 chromosomes. The 6 chromosomes which characterize the gender in males are XY, but in females are XX. Anyways, these spermatogonia divide by mitosis into daughter spermatogonia. There are 2 types of cell division. Mitosis, the father or the parent here are identical to the daughter. They either have diploid number of chromosomes, but the other type of division that we are going to see in the maturation, the daughter is having haploid number of chromosomes. The word haploid, just like diploid, describes the number of chromosomes. Haplo in Greek means single. So they have n set of chromosomes. In humans, this n equals the number 23. Okay, if you're not following with me, let us repeat in a quick way. We said the spermatogonia will proliferate and then grow to become a cell called spermatocyte. And that's it. The spermatocyte is first primary and then becomes secondary before changing its name to the spermatid. The word maturation here refers to the meiosis cell division, but proliferation refers to the mitosis cell division, and we already mentioned the difference. Now, where we are, we reached to the spermatocyte. Which one? The first one, the primary. The primary is changed into the secondary by meiosis, and then by another meiosis, it is changed into spermatid. And these 2 meiosis cell division together called the maturation stage. The first one is meiosis 1, the second one is meiosis 2. Because in microbiology, you are going to study meiosis in details, you will see or know that a cell in meiosis with the 2n diploid number of chromosomes will divide just like that into haploid number of chromosomes. This here is meiosis 1, and then divided again into 4 cells with another meiosis, the second part of meiosis 2. This having 1n, but the parent having 2n, meaning they are not identical. That's why they are called meiosis cell division. And comparison between meiosis and mitosis and the words haploid and diploid, these are going to be clarified later in the microbiology. Anyway, with our summary, let us return to the book. Here we have the maturation. We formed before in the previous stage the primary spermatocyte. The primary, which is the first, undergoes the first meiosis to form the secondary spermatocyte. And the secondary with its name undergoes the second meiosis. Here, secondary spermatocyte undergoes the second meiosis to form the spermatid. So all what we have to do is simply write the summary in front of you. And remember that first we have spermatogonia, followed by spermatocyte, then spermatid, then spermatozoa, and names to be remembered. And the meiosis, of course, at the end forms haploid number of chromosomes, and haplo, we said, means single in Latin, so it has 23 chromosomes. Here, if you notice, it's written 22 and plus 1. If it's Y, then it's male. If it's X, then it's female when it fertilizes the ovum. So the father defines the gender, not the mother. This note here, in terms of division videos, are less important, but we are going to witness the videos, the questions, and know if any asked here or not. And this diagram clarifies the stuff as well. We start by the spermatogonium, the singular, the plural spermatogonium. Or you can say the primordial germ cell, but this is less important. Then converts into primary spermatocyte, and the primary undergoes the first meiotic division to form the secondary spermatocyte, which undergoes the secondary meiotic division to form the spermatid. All are names to be remembered. And lastly, we form there the spermatozoa. Now, if you notice, these all cells are circular. They have circular shape. But the last one, the sperm itself, or the spermatozoa, to be more clear, has some characteristics that's needed to be mentioned. If we witnessed here till here, it has a head, then followed by neck, then followed by body, and lastly tail. These 4 parts are to be remembered. First, just like a person having a head, and any head is followed by a neck, then a body. And instead of legs, we have here tail for movement. These 2 having X, so they are males, are females when they fertilize with the ovum, forming XX, referring to females. And these 2 having Y when they fertilize the ovum, which always have X, will form XY, and this is a resemblance or resembling of males. Anyway, the transformation from the spermatid into spermatozoa has to occur in a special way, as the shape of the cell itself has changed. This process is called transformation, or its other name, spermiogenesis. And notice here, it's not called spermatogenesis, but with the I. It is the last stage of the spermatogenesis. The transformation simply lets the spermatid forms the spermatozoa. The T here, which I've written in the summary, refers to the transformation process. Here is your last chance to stop the video and write this summary, which collects all the information together. And don't forget this mnemonic also to remember the steps. The spermatozoa will be having the parts that we have just mentioned. It will be having a head and a neck, a body, and lastly tail. We should be knowing the spermatid or the previous stage cell contents to know which one will form these parts of the spermatozoa. The spermatid, just like any cell, will be having these organelles that you may have studied in the histology, which I didn't explain of this module if you're having it. A spermatid, just like any cell, having Golgi apparatus, nucleus, mitochondria, centrosome, and some other things. The Golgi apparatus, its function is producing enzymes. So it has to be converted to the spermatozoon in a part that has a similar function. Let us witness the sperm here. The Golgi apparatus, in fact, converts into the acrosomal cap. The word cap means it covers the head just like a cap, and acro means tip because it's at the tip of the sperm, and soma means body. So it's a body that's on the top of the head of the sperm. Its function is like the Golgi apparatus, forming enzymes to penetrate the ovum later. The nucleus converts into a nucleus as well of the spermatozoon. Mitochondria remains here in the body, which produce energy for the movement of the sperm. The neck contains centrioles that some axial filaments extend out of it to form the tail. I recommend to you to stop the page and this image to draw it on you right now on the paper that you are studying on. Here, just as we said, the neck or the head formed of the nucleus derived from the nucleus of the previous cell. The acrosomal cap, since it produces enzymes, it has to be derived from something that produces enzymes as well, the Golgi apparatus, to allow penetration of the ovum. The neck has two centrioles that give rise to axial filament that forms the tail for the motility or the movement of the sperm. In the middle, we have body. This body has mitochondria, and mitochondria, as we know, with its function, produce ATP that provide energy for the movement of the sperm to reach the ovum. And the site of the fertilization, the ampulla of the uterine tube, which we will know later, inshallah. Anyway, here there are some data or statistics to be noticed. We just need to remember the number 6. The sperms are 60 microns in length. 1 millimeter equals 1000 microns. So they are very short. And their production process needs 60 days to occur in their site of production, the seminiferous tubules of the testes. In each ejaculate during the intercourse, 600 million sperms are produced, but not all of them are active. Not all of them can move and are alive. The active sperms reach the site of the fertilization where it meets with the ovum 60 minutes after their deposition in the female genital tract. So that's to be considered if a female is going to take a contraceptive pill. To be noticing the timing has to be taken before the fertilization to occur. And the viability or the sperm remain alive to how many hours after deposition? Well, like any cell, it needs nutrition. If no nutrition, it will die. It cannot survive more than 48 hours. And this is important during race test, during raping test, in the medical legal book, which we'll be studying later, inshallah. So anyway, I tried what I can to deliver to you any information I know. And as you can notice, my English is not at its peak right now, but it has to be improving as time passes. So just follow with me. I'll try at utmost to make this module easy to you, inshallah, or others. وتوكلنا على الله.